Hip hip hooray. Hip hip hooray, it's time for hip hip hooray, it's time for SOSA. Bam! Hey everybody, hello, I'm Josh Starmer and welcome to the SOSA 2020 conference. I'm super excited to be here and I'm really glad you guys are here too. And if you're new to SOSA, SOSA is an organization that's very near and dear to my heart. They're doing awesome work and it's a total honor to be able to speak at this conference. If you're new and you're just visiting, I would like you to recommend or suggest that you do what I just did myself, which is go to the SOSA website. It's http://thesosa.org and donate some money. I mean, this is a great free thing. So if you've got a few extra dollars on your hand, it doesn't have to be a huge donation. You can donate with PayPal or a credit card. Anyways, just do what I just did and donate a little bit because this is an awesome thing. So with that said, let's get down to work. We are going to share a portion of my screen. And we're going to get rid of this, and we're going to share this. And there it is, bam! Machine learning, a microscope for genomics. So this workshop consists of 3 parts. Hold on, let me just move some stuff around on my screen. First, I will introduce a very simple machine learning method called a naive Bayes. Then I will provide a brief overview of a genomics technique called ChIP-seq. Then I will show you how to combine machine learning with genomics data to generate new insights. Note, machine learning refers to the techniques used for self-driving cars, identifying your friends in the pictures you take, and recommending TV shows. These fancy-sounding methods also work in a lot of other settings, including, as we will soon see, biology and genetics. So first, let's learn about a very simple machine learning method called naive Bayes. Imagine we received normal messages from friends and family. And we also received spam, unwanted messages that are usually scams or unsolicited advertisements. And we wanted to filter out the spam messages. So the first thing we do is make a histogram of all the words that occur in the normal messages from friends and family. We can use the histogram to calculate the probabilities of seeing each word, given that it was in a normal message. For example, the probability we see the word dear, given that we saw it in a normal message, is 8, the total number of times dear occurred in normal messages, divided by 17, the total number of words in all of the messages. And that gives us 0.47. Oops. So let's put that over the word dear so we don't forget it. Likewise, the probability that we see the word friend, given that we saw it in a normal message, is 5, the total number of times friend occurred in normal messages, divided by 17, the total number of words in all of the normal messages. And that gives us 0.29. So let's put that over the word friend so we don't forget. Likewise, the probability that we see the word lunch, given that it is in a normal message, is 0.18. And the probability that we see the word money, given that it is in a normal message, is 0.06. Now we can make a histogram of the words that occur in the spam. And we calculate the probability of seeing the word dear, given that we saw it in the spam. And that is 2, the number of times we saw dear in the spam, divided by 7, the total number of words in the spam. and that gives us 0.29. Likewise, we calculate the probability of seeing the remaining words given that they were in the spam. Bam! Now, because these histograms are taking up a lot of space, let's get rid of them, but keep the probabilities. Oh no, it's a terminology alert. Because we have calculated the probabilities of discrete, individual words, and not the probability of something continuous like weight or height, these probabilities are also called likelihoods. I mention this because some tutorials say these are probabilities and others say they are likelihoods. In this case, the terms are interchangeable, so don't sweat it. Now, imagine we got a new message that said, "Dear friend," and we want to decide if it is a normal message or spam. We start with an initial guess about the probability that any message, regardless of what it says, is a normal message. The guess can be any probability we want, but a common guess is estimated from the training data. For example, since 8 of the 12 messages are normal messages, our initial guess will be 0.67. So let's put that under the normal messages so we don't forget it. Oh no, it's another terminology alert. The initial guess that we observe a normal message is called a prior probability. Now we multiply that initial guess by the probability that the word dear occurs in a normal message, and the probability that the word friend occurs in a normal message. Now we just plug in the values that we worked out earlier and do the math. Beep, boop, beep. And we get 0.09. We can think of 0.09 as the score that dear friend gets if it is a normal message. However, technically, it is proportional to the probability that the message is normal, given that it says dear friend. So let's put that on top of the normal messages so we don't forget. Now, just like we did before, we start with an initial guess about the probability that any message, regardless of what it says, is spam. And just like before, the guess can be any probability we want, but a common guess is estimated from the training data. And since 4 of the 12 messages are spam, our initial guess will be 0.33. So let's put that under the spam so we don't forget it. Now we multiply that initial guess by the probability that the word dear occurs in spam, and the probability that the word friend occurs in spam. Now we just plug in the values that we worked out earlier and do the math. Beep, boop, boop. And we get 0.01. Like before, we can think of 0.01 as the score that dear friend gets if it is spam. However, technically, it is proportional to the probability that the message is spam, given that it says dear friend. And because the score we got for normal message, 0.09, is greater than the score we got for spam, 0.01, we will decide that dear friend is a normal message. Double bam! Now, before we move on to a slightly more complex situation, let's review what we've done so far. We started with histograms of all the words in the normal messages and all the words in the spam. Then we calculated the probabilities of seeing each word, given that we saw the word in either a normal message or spam. Then we made an initial guess about the probability of seeing a normal message. This guess can be anything between 0 and 1, but we based ours on the classifications in the training dataset. Then we made the same sort of guess about the probability of seeing spam. Then we multiplied our initial guess that the message was normal by the probabilities of seeing the words dear and friend, given that the message was normal. Then we multiplied our initial guess that the message was spam by the probabilities of seeing the words dear and friend, given that the message was spam. Then we did the math and decided that dear friend was a normal message because 0.09 is greater than 0.01. Now that we understand the basics of how naive Bayes classification works, let's look at a slightly more complicated example. This time, let's try to classify this message. So it says lunch, money, 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 money. Note, this message contains the word money 4 times. And since the probability of seeing the word money is much higher in spam than in normal messages, then it seems reasonable to predict that this message will end up being spam. So let's do the math. Calculating the score for a normal message works just like before. We start with the guess, then we multiply it by the probability we see lunch, given that it is in a normal message, and the probability we see money 4 times, given that it is in a normal message. By the way, I'm using a shorthand for the probability of seeing the word money 4 times. I've got it raised to the 4th power. If I didn't want to use an exponent, I could just say times the probability of money, given that it's normal, times the probability that it says money, given that it's normal, times the probability that it says money, given that it's normal, times the probability that it says money, given that it's normal. The only reason why I'm not doing that is it would run right off the edge of the screen and we wouldn't be able to see it. So I'm using this exponent of 4 to sort of make the math a little smaller. Okay, that being said, when we do the math, we get this tiny number, and that's kind of what we expected. However, when we do the same calculation for spam, we get 0, and that's not what we expected. This is because the probability we see lunch in spam is 0, since it was not in the training data. And when we plug in 0 for the probability we see the word lunch, given that it's in a spam message, then it doesn't matter what value we picked for the initial guess that the message was spam. And it doesn't matter what the probability is that we see money, given that the message was spam. Because anything times 0 is 0. In other words, if a message contains the word lunch, it will never be classified as spam. And that means we will always classify the messages with lunch in them as normal, no matter how many times we see the word money. And that's a problem. To work around this problem, people usually add one count, represented by a black box, to each word in the histograms. Note, the number of counts we add to each word is typically referred to with the Greek letter alpha. In this case, alpha equals 1, but we could have set it to anything. Anyway, now when we calculate the probabilities of observing each word, we will never get 0. For example, the probability of seeing lunch, given that it is in spam, is 1 divided by 7, the total number of words in spam, plus 4, the extra counts that we added. And that gives us 0.09. Note, adding counts, those black boxes, to each word does not change our initial guess that a message is normal or the initial guess that the message is spam. Because adding a count to each word did not change the number of messages in the training dataset that are normal, or the number of messages that are spam. Now when we calculate the scores for this message, we still get a small number for the normal message. But now when we calculate the value for spam, we get a value that's greater than 0. And since the value for spam is greater than the one for a normal message, we classify the message as spam. Spam. Now that we know how naive Bayes works, let's talk about ChIP-seq. Imagine we have a bunch of cells, and inside each cell there's a nucleus. And inside each nucleus there are chromosomes. Let's focus on the chromosomes. Specifically, let's focus on these chromosomes. Chromosomes are made out of chromatin. Chromatin is made out of DNA plus histones, which are a type of protein, plus other proteins that we'll talk about some other day. DNA wraps around the histones to package the DNA, and the packaging can regulate gene expression. Depending on how they are modified, histones can activate or repress genes. Lots more can be said about chromatin and how it's packaged, but that's for another day. Today, we just need to know that chromatin is essentially DNA wrapped around histones. To simplify things, let's just use this big blue line to represent chromatin from here on out, but remembering that it's made of DNA and histone proteins. And let's let these big brown arrows represent genes. And let this green circle represent a histone that has been modified to promote transcription. And let this red stop sign represent a histone that has been modified to repress transcription. In a cell, all kinds of proteins can bind to DNA. This mustard-colored thing might promote transcription of this gene, and this pinkish color thing might repress this gene. And who knows what this green thing is doing? We can use ChIP-seq to find out. ChIP-seq stands for chromatin immunoprecipitation combined with high throughput sequencing. And that's a mouthful, but don't worry about it. We're going to go through it one step at a time. In short, ChIP-seq identifies the locations in the genome bound by proteins. That's the most important thing about ChIP-seq. So I'm going to say it again, and I'm going to say it in bold. ChIP-seq identifies the locations in the genome bound by proteins. For example, say like we wanted to identify all the regions in the genome bound by the green thing. The first thing we do is use formaldehyde or something like it to glue all of the proteins bound to the DNA together with the DNA. This means that all of the DNA-bound proteins, including ones we're not interested in, are glued to the DNA. So everything is glued to the DNA. The next thing we do is cut the DNA up into small, approximately 300 base pair fragments. Boo! Then we isolate the protein we're interested in. In this case, we're interested in the green thing using an antibody. The black star represents an antibody that is attached to a bead. Then we isolate the proteins bound by the antibody and wash everything else away. Then we reverse the formaldehyde glue by warming up everything. Then we isolate the DNA by washing away the proteins. Now that we see how to isolate DNA that is bound by a particular protein, let's take a step back. So far, the example has focused on just these chromosomes. But the process, glue proteins to DNA, cut up DNA, bind proteins of interest with antibodies, isolate the antibodies, unglue and wash away proteins, applies to all of the chromosomes in the cell. And usually it is applied to a pool of about 6 million cells, give or take a few. So in the end, we end up with a lot of DNA fragments from a lot of cells. Then we prepare a sequencing library by adding sequencing adapters to both ends of the DNA fragments. Then we PCR amplify the library, check the library concentration, and then sequence. And lastly, we align the high quality reads to a genome. If this is the genome, and we just have 3 chromosomes in this small genome, then the first read might come from here, which is a location on chromosome 2. And the second read might come from here, which is a location on chromosome 1. And the third read might come from here, which is another location on chromosome 1, etc., etc., etc. Ultimately, we get a long list of genomic coordinates for all of the reads, usually between 50 and 100 million reads. Although this list is way too long to go through by hand, we can visualize it with something called a Genome Browser track. This specific genome browser contains gene and chromosome positions in the mouse genome. At the top, we have genomic coordinates. That's the chromosome name and the positions along the chromosome. Then we have schematics of the genes in that genomic region. Below, we have a track that we created from the 50 to 100 million ChIP-seq reads. A lot of reads mapped to this region, and relatively few reads mapped to these other regions. This track was made from a control experiment. The control track was made from taking some of the input chromatin from the original ChIP-seq experiment, and without using an antibody to enrich for any particular protein, ungluing all of the proteins and washing them off. Then sequencing, aligning, and converting into a track. In summary, the control track uses some of the same input chromatin for the ChIP-seq experiment, but doesn't try to enrich for any particular protein binding. We then use the control track to verify that a high concentration of reads in the ChIP-seq track is due to a protein binding there and not because a lot of reads mapped to a repetitive region. If you're not familiar with repetitive regions, they're just regions in the genome that are kind of sprinkled out all over the place. And if a protein maps to one location, it's likely all kinds of stuff's going to map to that location because they're repetitive and there's just lots of copies of them. Okay, statistically significant peaks are usually represented on genome tracks by rectangular bars. We can then compare peaks for the same protein in different cell types, like lung versus stomach. Or, if we didn't know the specific DNA sequence that this green thing bound to, we could guess that it is a motif found in all of the peaks. So this green thing might bind to this DNA motif, which has a lot of G in it. We can also try to determine the functional role of the gene of the green thing by looking at where it binds relative to the genes. Here, we see that the green thing binds near the start of a gene, so it might regulate that gene in some way. These are some of the things you can do with ChIP-seq. In summary, ChIP-seq identifies the locations in the genome bound by proteins. Double bam! Now that we understand the main ideas behind naive Bayes, which is a machine learning method, and ChIP-seq, which is a genomic technique, let's talk about how we can combine them to gain new insights about genes. Now, going back to this big blue line that represented chromatin, remember these big brown arrows represented genes. And this green circle represented a histone that was modified to promote transcription. And this red stop sign represented a histone that was modified to repress transcription. Now, in this example, it's pretty obvious that this gene could be transcribed because it has a modified histone that promotes transcription. And it's pretty obvious that this gene may not be transcribed because it has a histone that inhibits transcription. Unfortunately, in real life, the situation is a little more complex. Instead of just having two easy to interpret histone modifications at different genes, we frequently see them mixed up. And it turns out there are a lot of different modifications. And it's not always clear if they promote or inhibit transcription. That said, if we had a few genes that we knew were active and a few genes that we knew were inactive, then we can treat histone modifications like words and create a histogram of how frequently they occur. And now, when we get histone data for a new gene and we don't know if it is active or not, we can use naive Bayes to find out. Now that we know the theory of how this might be possible, let's look at the steps required to make this happen. First, we need some data. When you don't have your own data, and that's me because I don't have access to data, one great resource is a website called ENCODE, which has a lot of RNA-seq and ChIP-seq data that you can download for free. So in this example, all of my data came from ENCODE. For this demonstration, we will classify genes as active and inactive using RNA-seq data from ENCODE, which is like ChIP-seq for active genes. Then we will merge the gene data with ChIP-seq data for the histone modifications. Then we will take some of the active genes and some of the inactive genes and put them to the side to form something called a test data set. Ultimately, we will use the test data to evaluate how well the machine learning method performed. Now, with the remaining active and inactive genes, we create a naive Bayes classifier. Then we run the test data through the naive Bayes classifier to evaluate how well the classifier performs. Bam! Note, while this example is not exactly groundbreaking, the point is to demonstrate an approach that can be easily applied to all kinds of other situations in genomics. Now let's look at the code. So give me just 1 second I got to share a different screen. I'm going to share this screen. Okay. So what we're looking at is something called a Jupyter Notebook. Um, this is for Python. And by the way, I've got code for Python and R. So if you are familiar with Python or you just want to learn how to code in Python, I've got one of these documents for Python. And if you're familiar with R or you want to learn how to program in the language R, then I have a document for that as well. And they're basically the same. Both of them are annotated and have descriptions of everything we need to do. They also include links to the original raw data. So if you want to learn more about the OnCode project, you can just click on this link and it'll take you to the OnCode website. And if you want to learn more about the data that we collected, you can click on these other links and you can click on these links to access the data directly. And then below that, I've got descriptions of what we need to do and why we need to do it to create a naive Bayes classifier that can predict whether or not a gene is active or inactive based on histone modifications. And so what we have is we've got a nice description of what the first step is. And then below that, we actually have the code. And we can run that code right here in the browser by clicking run selected cells, or there's a keyboard combination that we can type, or we can press this play button and it'll run that code. And once you see a number there, it means that the code is run. Now we can go to the next section. So there's a big long description of what we're doing and why we're doing it. And then there's more code that we can run. So we'll run that. We're going to read in our data set. And now we're going to print out the first 5 rows. And basically, this Jupyter Notebook goes one step at a time, explaining every little detail of what we're doing and why we're doing it. And one thing you'll notice about machine learning and data science is that even though those sound fancy, if someone says, I'm a machine learning engineer, you're like, wow, that sounds like you do something super fancy. 99% of this notebook is describes how to format the data. It's not until we get all the way down here, right here, these 4 lines of code, this is the machine learning part. Just because I'm a big believer in spoilers and ruining the fun, what I'm going to do is I'm going to run all of the code above this. By the way, I don't know if I mentioned this, all of this is going to be downloadable for free, and we'll send out a link to the URL when we send out sort of the post-meeting information. So you can all download this and you can do this yourself. So what I did is I just ran all of the code above. We draw a nice graph or whatever, but we haven't run this. This is the machine learning part right here. This is our naive Bayes when we create it, and we're going to test it. And here it is. So let's run it. Bam, we just ran it, and it actually does okay. We correctly identified whether or not 82% of the genes are active or inactive. And like I said, I also have the same code for R. So if you're interested in learning R, or you already know R, you'll have this file that can go through, and once again, it explains every little step, what we're doing, and why we're doing it. And so you can get the exact same thing done in R. And like I said, this is all going to be downloadable for free to people that attended this conference. So yay for you. Hooray. Okay, so that's the end of my talk. I'm available for questions if anyone would like to ask them. Daniel, I don't know if you could moderate those, or there may be no questions. I can always just play more ukulele songs if I have to. You are muted.
Sorry, I'm not seeing any questions in the chat box for right now.
That's totally fine.
Maybe you can just type them in the chat. Or if you want to ask me, I can just read them out.
That's totally fine. 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 That's tot That's totally fine.
I
Right.
might have a quick question for you, Joshua.
Sure.
For when you integrate encode data into your machine learning approach, did you restrict this to just one cell type, or did you go beyond that? And can you elaborate a little bit how that would affect the prediction by
Yes.
going to different cell type?
Yes. So what I specifically, I used K562 cells. Um, and so so that what that means is there's only one cell type. And that's a very cryptic name for a type of human leukemia cell. Um, so yeah, so what if you want to use different cells? Say like I wanted to use, because what's cool about OnCode is you can get cells for different tissues. You could get cells for pancreas or liver. Um, and say like we wanted to compare and contrast, or maybe we could, based on certain things, can we predict whether or not that gene is being transcribed in liver or pancreas or something like that. Um, so there's a lot of implications. So of what that would mean if we use different cell types, our prediction. I mean, we could use them in different way. For example, like I was saying, we can predict whether that gene is being transcribed in one cell versus another, or cell type, excuse me, versus another. Um, it also would make our machine learning algorithm more robust if we added additional cell lines or additional tissues. In that, right now, if we want to make a prediction, it's probably a good idea that question, excuse me, comes from data that is generated from K562 cells. However, if we trained using multiple cell lines, we could say, who cares where the data comes from? We've trained it on so many, we've seen a lot of different variations for each gene. It might be more robust and better at making predictions in a wide variety of cell types and tissue types. So that would make it, the technical term for that is it would make it more robust. So that's what would happen if we added more cell types and tissue types to our machine learning process.
Okay, thanks. Um,
Yeah.
we do have some questions here. This is from Harry Slam. Did you have to tune any parameters for the naive Bayes classifier for these genomes?
Yeah, so for this example, to keep it as simple as possible, I did not do any tuning. This is totally off the shelf. I used all the default settings um, for the naive Bayes classifier. Hold on, let me just scroll down and look at that code just to verify. Yeah, I used all the default settings. Here, I'll just show you. Let me show you right here. Bam. So you'll see we've got 4 lines. This is the line that creates sort of an empty shell of a naive Bayes classifier. And you'll see within these parentheses, I've got nothing. So I'm not specifying any fancy non-default tuning parameters. Here's where I fit or train the model using the training data. And again, there's no special training parameters in there as well. And then basically this line, what this does is it just makes this confusion matrix look a little bigger. And then we plot this thing called a confusion matrix, which basically tells us how well our predictions are. So on the y-axis, we've got the true label, whether or not a gene was not active or active. And then we have the predicted label, not active and active. And you see that most of the genes fall in the diagonal, meaning they were correctly predicted. And that was with no, in machine learning, we could say, there was no hyperparameter tuning or anything like that. We could get all fancy in that, but I didn't do any of that with this stuff. I just took it right off the shelf and used it, and we got 82% accuracy, and that's pretty cool. So you don't have to be an expert in this to at least get started.